Hello everyone, thank you for watching our video. We're going to start off with a couple introductions of who our Group 2 members are. We have Gabrielle, a undergraduate student from the Universidad San Francisco de Quito.
Hello everyone, my name is Icarus Montesi and I am a master's student from USP, Brazil.
Hello everyone, my name is Mateus and I'm pursuing my master's degree in the Plant Genetics Improvement Program at Exalc USP in Piracicaba.
Hi, my name is Paige. I'm a fourth year environmental science major specializing in climate change and air quality.
Hello, my name is Zach Jaramillo. I'm a UC Davis PhD student. The three plants our group will discuss is first, a food crop from the United States, the blueberry. Second, for wild/ecological importance, is the tagua palm from Ecuador. Third plant is Erva cidreira, a medicinal plant from Brazil.
Okay, I'm going to give a brief introduction of blueberries in the United States. Their scientific name is Vaccinium cyanocus. The plant type is a perennial flowering plant. Geography, they are native to North America, specifically the US, and grown in 26 states. The climate is humid, northern, and where there's winter chills, midsummers, and low pH or acidic soils. The growth period is between 6 to 8 years before they reach full potential and production. Health benefits, they have antioxidants, vitamin C, folate, etc. History of cultivation. In the early 1900s, the first commercial crop of blueberries was grown, and then today there's around 1 billion pounds of blueberries that are grown for consumption. On the next slide, we have a couple photos that show, on the left, it shows the growth stages going from the immature green all the way to the R ripe. And then in the middle, these are flowers on a cultivated blueberry bush. And then lastly, on the right, there's a blueberry farm in Washington State.
So here in this photo, we have a couple photos of my family picking wild blueberries in New England. A couple issues and challenges that blueberries face is one, the breeding and crop development is slowed due to the perennial nature of the crop. Additionally, post-harvest, there's different diseases that affect the quality and shelf life of the berries. Here we show a depiction of where blueberries are primarily grown in the US. And one of the key things that breeders are trying to develop into the crop are they're trying to expand where blueberries can be grown, and they're doing that by increasing cold tolerance, allowing for low chill tolerance, and then additionally, a key thing about blueberry bushes is typically they prefer more acidic soils, and so breeders are also breeding for tolerance of high soil pH. Additionally, some other key traits that breeders are focusing on are traits that would allow the use of mechanical harvesting. As you can see in these photos, oftentimes, especially for short bush blueberries, the harvest is manual, which can be very expensive. Likewise, another key trait that they're focusing on is shortening the ripening period so that they have a larger window of harvest and growing. Additionally, post-harvest, blueberries are susceptible to gray molds, fruit putrefaction, and in general, both of these diseases affect the quality and appearance of the fruit. Nobody wants to eat a moldy blueberry. And so typically both chemicals and fungicides are used to help prevent the disease, as well as different storing protocols are used as well. So for our potential research, one of the key things that we want to address is the crop development and breeding of blueberries. And so historically, blueberries have used traditional methods in breeding, and this is rather slow and can take anywhere from 9 to 20 years for a new cultivar to develop. And so within the last decade, there has been significant gains made in order to optimize both agrobacterium and CRISPR-Cas9 transformation systems into blueberry. And so we would like to build upon some of that work. And so additionally, when it comes to blueberries, blueberries have a genome that has been uploaded for both wild and cultivated lines. And within that, it has been discovered that cultivated lines are tetra or hexaploid, but across the wild species of blueberries, there's both diploid, tetraploid, hexaploid, and even a pentaploid species found. And this is important because both are within the traditional practices of breeding and crop development, wild species have been introduced in order to introduce traits into the developed cultivars. So our proposed research is a GWAS analysis of northern and southern high bush blueberries. And the focus for this GWAS analysis is identifying different QTLs related to pathogen resistance, high pH tolerance, and cold tolerance, because these are desirable traits that we want to introduce into the cultivars. And we're hopeful that this GWAS analysis will be fruitful because previous analysis on blueberries have identified different QTLs all related to different berry traits, whether that be fruit firmness, fruit texture, and flavor as well. So the goals of our GWAS analysis are to identify these candidate genes. We would follow up with validating these candidate genes. And then once we have our list of genes, we would seek to transform blueberries either through agrobacterium or CRISPR-Cas9 in order to help develop the crop and introduce favorable phenotypes.
Itelephas equatoralis, known as tawa, is a palm tree that reaches up to 2 meters in height. Its main product is tawa, which is a hard and fibrous material resembling ivory. This is an important resource in Ecuador because it is used in local craftsmanship to create various products, which implies cultural, economic, and environmental value. Among its secondary metabolites, it produces lignin, starch, fats and oils, tannins, and phytosterols. Some of the main problems facing tawa include overexploitation. The excessive harvesting of tawa can lead to depletion of tawa palm population, threatening its sustainability and the ecosystem it supports. Habitat loss, deforestation, and land conservation from agriculture, urbanization can result in the loss of the tawa palm habits, reducing its natural range and disrupting ecosystem. And the last one, the lack of regulation.
I will be presenting you our medicinal plant from Brazil, called Erva cidreira. Its scientific name is Lipia alba, and it's a plant from the family Verbenaceae. It's an aromatic shrub, and it's widely distributed in the Americas. The parts of the plant that are used are the leaves, the branches, and the inflorescences. And in the ethnopharmacological field, it's most famous for its calming activity, but it's also known for the analgesic, spasmodic, anxiolytic, and other purposes as well. Something very interesting that happens in this species is that it can vary its genotype because it's a polyploid organism. So it can vary also the metabolites, the chemical metabolites that can be found in that plant. And it's described in the literature some chemotypes that can be found according to those variations in the chemical inside this plant. For example, we have the citromyrcen and the citrolimonene chemotypes that are very similar between themselves, and they are known to the calming activity as described before. But we also have the carbon-limonene chemotype that is known for its pesticide activity. And we also have the linalool chemotype that is more related to the aromas and perfumes. But um, a problem that happens is that sometimes the morphological difference of those chemotypes is not so well noticeable as it happens to the medicine applications. For example, the chemotype citrullimonene and the chemotype carbonlimonene are very similar among themselves in a morphological perspective. And it's very bad if you think that those plants are used in a medicinal purpose and maybe they are not being correctly addressed, as it's very hard to differentiate those chemotypes. And also thinking in a conservation perspective, it's good to know more and generate the genetic resource about this species.
So for Erva cidreira or Lipia alba, our proposal project aims to identify genomic differences between the citrullimonene and carbonlimonene chemotypes associated with the differences in their essential oil compositions, and pinpoint candidate genes involved in the biosynthetic pathways of their main metabolites, since they are morphologically similar but differ in their chemical content and applications. First of all, it's important to note that Erva cidreira has a reference genome in the NCBI that was recently uploaded. However, this genome was sequenced only with short reads, and there is no information about the respective chemotype or genomic annotation available. Therefore, it would be suitable to select individuals from both chemotypes of interest for genomic sequencing using different short and long-read sequencing platforms to ensure comprehensive coverage and accurate genome assembly. Subsequently, bioinformatics processes will be employed for de novo genome assembly, annotation, and comparative analysis, including the identification of SNPs and alleles potentially linked to the biosynthetic pathways of characteristic metabolites in each chemotype. This comprehensive approach would facilitate the identification of specific variations between the chemotypes. Going further, we could seek out for potential genes responsible for producing those metabolites of interest. This would involve an additional layer of investigation to spot differences between the chemotypes, focusing on transcription. As there is no existing reference transcriptome, we will need to create one that encompasses the full range of transcripts in the species. Following this, we will collect leaf samples from individuals of both chemotypes for RNA-seq, since this is the organ generally used for its medicinal properties, and then we could predict the most prevalent transcripts in the leaves of each chemotype, confirming its differential transcription, and pinpoint the most active metabolic pathways in each chemotype. Furthermore, it will be important to identify and quantify the metabolites produced by each chemotype. This would offer further clarity into the biosynthetic pathways. Metabolomic analyses are well suited for this task as they enable the identification of pathway products and facilitate comparisons. To achieve this, leaf samples from individuals of both chemotypes could be collected and employed in the analysis. Then, through the integration of omics data generated, it will be possible to produce a holistic view to understand the complex biological pathways between the chemotypes. Computational analysis would be conducted to identify relationships between the different omics datasets, providing insights to identify candidate genes involved in specific biosynthetic pathways of each chemotype. Subsequently, the pathways could be reconstructed and tested in a heterologous system for validation and/or production of a compound of interest.
We'd like to thank you for listening to our presentation. Final words, goodbye, see ya, thank you, obrigado, gracias, ciao.